Good afternoon, and thank you for joining us for today's EMSL Learn webinar, where we will be talking about high-resolution capabilities to advance research in critical minerals behavior. Our next EMSL Learn webinar is on June 25th, where we will be continuing the topic of critical minerals and discussing EMSL's capabilities for chemical and biological characterization. More information will be posted on EMSL's social media channels and provided through our email. If you aren't signed up for our newsletter, this is a great reminder to subscribe. We're going to be posting some links for you where you can access our newsletter. Today's presenters, we have 4 for you today that will be talking about our capabilities for high-resolution imaging, including Odeta Qafoku, an EMSL Earth scientist who will discuss electron microscopy, Jeremy Bougoure, an EMSL chemist who will discuss nanosecondary ion mass spectrometry, Danny Perea, an EMSL materials scientist who will discuss atom probe tomography, and Arunima Bhattacharjee, an EMSL Earth scientist who will discuss EMSL's suite of synthetic habitat capabilities, which we call TerraForms. The last 15 minutes of our webinar today will be dedicated to questions. We invite you to use the chat or the Q&A section of Zoom to post your questions, and our presenters will answer them then. And with that, I'd like to invite Odeta Qafoku to kick off our webinar.
Hello, everybody. Just moving along. Okay, great. Thanks, Jenna, for the introduction. I'm Odeta Qafoku. I'm a soil and geochemist here in EMSL, and today I would like to talk about electron microscopy as a high-resolution capability for critical mineral research. But before I start talking about electron microscopy, I just want to take just a second here to talk about the critical minerals or critical elements. And if you are looking at this slide and if you are looking at the graphs, which I think a lot of people are familiar with, you will see a number of metals and elements that are in this right corner. And those are what are considered critical because they are at high demand for energy and technology and defense sectors, but they are at low supply, or they are hard to get, or they are at low concentration. So that's why EMSL Exploratory is focused on research with different users on this field. And where are those critical minerals? They are everywhere in a lot of places in the United States. If you look at the USGS map, you will see where they are concentrated. And I'm talking here primarily about the domestic supply. They are primarily in mineral ores, but they also are in mine tailings. They are in soils at lower concentration. They are in marine sediments. So in this seminar, I would like to show our capabilities and offer the opportunities to use our high-resolution instrumentation to address questions of where, how, and in what form are these critical minerals in the research fields that you are interested in. So okay, it went too far. I'm sorry about that. I need to back one slide. All right. So I just want to mention here that EMSL is very, very well equipped to address the critical mineral research. We have a number of high-resolution capabilities. They are shown in here, and I think we have more, and I have not probably covered everything. But what I've covered here are the capabilities that we will talk today and will describe today, and those are all highlighted in green, and a number of capabilities that our scientists will discuss in the next webinar. So with that completed, I would like to start moving now to talking a little bit about electron microscopy. And I know that this is an instrumentation that many people might be familiar with, so I don't want to go too much in detail, but I just would like to have in a nutshell, what this instrument is about. You have an electron beam that you can activate and generate using the high voltage and high current, and then through a number of lenses, you can direct this electron beam to go and interact with the sample. As it interacts with the sample, it generates electrons and it generates X-rays that, depending on what detector you use to collect those, you can get a lot of information about sample characteristics. So if you collect the secondary electrons, those are low energy electrons, and they are ideal to give information about the morphology of the sample. If you could collect backscatter electrons, because they are formed as a result of interaction of the electron beam with the atom of the elements that you are interested in, you get a lot of information about composition of that sample, and I'll show example of what I mean by that in a little bit. If you collect the characteristic X-ray using the EDS detector, then you get information about composition of your sample. And lastly, if you use the focused ion beam, which instead of electrons, they use gallium ions, which are high energy, you can cut through your sample, you can look at the interfaces, and also you can prepare a sample that can be extracted and can be prepared for transition electron microscopy analysis. So I'll go through some examples of how these different type of detectors can help with critical mineral research. And some of the examples that I will show here are from the literature, and some examples are from data that we've collected here. I'm starting with images that are collected using secondary electrons. And in this example, especially in the one on the left, you can see a lithium-bearing rock that on the top is of the alpha form, and on the bottom is a beta form. And you can see that the changes that are happening in the structure of this rock by just heating it at 1000 degrees C. You can see the delamination of the rock, and that is a very good way of extraction of lithium out of this rock-bearing mineral. Lithium is one of the critical elements, so just looking at the morphology gives you some information of strategies of how to extract this critical element from this material. Similar to that, if you look on the right, you have two examples that shows a dynamic crack that is formed at different loadings. Those cracks are formed to liberate the critical minerals that are contained in the top rock, which is a sandstone, and in the bottom one, which is gabbro. They both are known to contain critical minerals. Again, this is a strategy to see if the protocol that you're using is working or not. Moving along with backscattering electron detector images, those images show information about the composition of your elements of interest. And that is done through these high contrast areas, because this backscattering electron gives information about the atomic number of the elements that you're interested. The higher the contrast, as you can see in some areas we have high contrast, that is indicative of presence of elements that have high atomic number. And in this case, we have the high contrast area that contains germanium and zinc, but also it's interesting to see some other areas that are also slightly enhanced in contrast, and they appear to be as a rim around the particles, and in that case, those contain germanium also. If we look at the image that I'm showing on the right, this is a rock that has been exposed to extraction fluid. And what this indicates, first indicates that you have a cocoon here formed with a mineral that has not been extracted at all. And also another important thing to observe here is you have some zonal layering that are shown in this image where copper and iron are alternating concentration. Again, those information from those images gives you ideas of whether your extraction protocol for releasing the liberating the critical minerals is working or not, and develop new strategies. Here I'm showing data that is collected using energy dispersive X-ray spectroscopy. I would say that this application is one that has been developed tremendously in the last 5 years. Now we are in a point where we can do fully automated analysis, and we can do a lot of AI, machine learning, extraction of our data. So first, let's focus on the image that I have on the far left. And in this case, this is a chemical image, a chemical composition of a rock. And you can see how I not only have looked at composition, but through AI-derived machine learning, I have also identified phase ID. And here I'm identifying iron, titanium oxide, and apatite. Both of those minerals are known to contain rare critical minerals. So knowing how they are spatially distributed, it's important to decide the strategies of the extraction. In the image in the middle now, I'm showing another data type, and this is collected now of soils, because I just mentioned that the critical minerals don't only happen in rocks and in or deposit, but also in soil. And in this case, the data that I'm showing here is collected by Elias Nakuze. He is a staff scientist here in PNNL, and he not only collected the data with EDS detector, but he also did some correlation analysis to see correlation between manganese and iron in this example. The third example that I have here, which is showing how rare earth actually are distributed in red mud. The data here is collected by Qin Zheng. He is also a staff scientist here in PNNL, and you can see that the rare earth actually are within grain boundaries rather than in the grain itself. The last example that I'm going to show here is using transmission electron microscopy. I did mention that we can extract a lamella that is very thin, between 50 to 100 nanometers. In this case, the lamella is extracted from this zone here. And what we wanted to understand here is difference in composition at this zonal layering. And what we found by using transmission electron microscopy, and specifically I'll call here Libor Kovarik, which is our staff scientist that uses transmission electron microscope, you can see much more information when you do mapping of that very thin section. And he also did some diffraction characterization of different phases, and he found a mineralogy that is present in the sample as a result of redox-driven reactions. The last example I also show here is if you want to focus on the C insert, this is from a soil, and this is kind of very interesting. Again, the data is collected using transmission electron microscope on a very thin lamella. You can see that the zinc in this sample is associated with a silicate, while copper, very interesting, is associated with organic matter. Again, all these are information that decide on strategies that you could use for extraction of critical minerals from these different deposits. So I would like to summarize here my presentation about electron microscopy. Again, it's a very versatile technique, and it can use for all kind of characterization of critical mineral research, and would like to do this in collaboration with all EMSL users. So I would like now to introduce Jeremy Bougoure and give this mic to him to talk about the nanosims.
Thanks, Odeta. So I'm going to take a change of tact now and talk about secondary ion mass spectrometry. Holly, could you move the slides for me? Sorry, my controls aren't working. Thanks, Holly. So the secondary ion mass spectrometry, or SIMS as we like to call it, is a solid surface analysis technique. There are quite a few variants of the technique, but the simplest and most general description starts with the primary source of ions, what I'd call a beam, being focused onto the sample surface. As this primary beam sputters the surface, secondary ions are ejected from the sample. It's these secondary ions that are separated by one of a few different methods, by something like time of flight or magnetic sector analyzer, before being counted on something like an electron multiplier or a Faraday cup. Obviously, this is a very simplified description of SIMS, and it's worth noting that there are considerable configuration variations between instruments, which result in various strengths and weaknesses between them. So why do I or you want to actually use SIMS? Well, firstly, SIMS analysis is renowned for high sensitivity, meaning concentrations of elements or ions of interest at parts per million and even parts per billion are often ample for analysis. Collectively, SIMS instruments have pretty decent resolution or spot size, and there's generally a good instrument fit dependent on the specific scientific questions being explored, with beam size as low as the tens of nanometers for some instruments. So what can we measure with SIMS? Generally, anything from single isotopes to small compounds, again, with the choice of instrument dictating specific potential. For example, more ubiquitously available TOF SIMS systems are better suited to molecule and element characterization, whereas dynamic SIMS systems, such as the nanosims, are far better at assessing isotope chemistry of samples. Because of the limited time here, I'll only concentrate on the SIMS variant, the nanosims, that I'm most familiar with, and look after here at EMSL. Next slide, Ollie. So the nanosims has been optimized for high resolution, high sensitivity, and high mass resolving power, and is typically considered an excellent choice for imaging and mapping isotopes across the entire periodic table, including those critical minerals that Odeta described earlier. As with other SIMS instruments, a primary ion beam bombards a sample surface, and the ejected secondary ions are sorted based on mass charge and measured simultaneously by 7 position detectors. The beam can be rastered across sample surfaces with collected data points combined to generate images, like you can see on the screen at the bottom left. Individual images from individual detectors can then be overlaid to generate isotopic information for a single element. For example, we could calculate the spatial carbon isotope concentration patterns of a sample, like is shown for the microbial cells on screen. Or we could overlay data for different elements to better understand their spatial patterning as well. So like in the example on screen, we can see carbon isotope variation amongst the image microbial cells. We can also see other spatial information as well. In this instance, we've also measured sulfur, chlorine, and aluminium oxide. But the reality is we could interchange any of these isotopes and elements depending on the scientific questions being targeted. Importantly, any of the critical minerals and their isotopes could quite easily be analysed in such a way. Okay, the next slide, Holly. By now, you may be thinking that this technique might just work for your research, and that's great. But how do I make this happen? How do I get my sample from the soil or an organism or even the petri dish into the instrument and start getting some nice data? So the three basic principles of sample preparation are that the samples need to be flat, able to withstand high vacuum, and be conductive. Obviously, there are some tried and proven ways of creating such a sample, but there is no one-size-fits-all approach. Sample preparation may be as simple as pipetting bacterial cells onto a conductive sample holder, polishing a mineral particle, all the way through to more complex cryo-ultramicrotomy of free substituted environmental samples. The specific aims of your research and sample characteristics will determine how samples are prepared. And as in the case for many techniques, it's this sample preparation that is the key to obtaining good data. So the image on screen here is a top-down view of some of our sample holders and alludes to the variety of samples we can accommodate, including those on metal stubs, flat top resin stubs, sections or small particles on flat surfaces such as EM grids, silicon wafers, even stuck down filters, or even glass slides. As long as samples are dry, flat, and conductive, they will likely be fine for analysis. Can I get the next slide, Holly? So in the following few slides, I'll briefly discuss the application of nanosims analysis to a few actual and potential critical mineral-focused areas of interest. So most existing nanosims analysis associated with critical minerals has focused on mineralogy and geology. It's obviously a good marriage for the technique. I'm from Western Australia, where mining is a big deal, including for many of these critical minerals. So it's something that I've heard much of in the past. Although nanosims is not a commonly employed analysis method for these types of samples, there is existing work that clearly demonstrates the potential of the technique. Most importantly, the technique works on these sample types and can detect any of the critical minerals at low concentrations and high resolutions. For example, the mineral sample on the right of screen, first, in the top left of that image, we have a backscattered electron image of a polished surface. Below that, the authors have overlaid data for iron, copper, and calcium to demonstrate the spatial relationship of those three on the same sample. The authors then reanalyzed the same area and collected data for strontium, neodymium, thulium, and thorium. In this instance, I don't so much care about the specific iron species, but really want to highlight the potential that any elements, including critical minerals, and even their isotopes, can easily be measured in a similar way. The images on the left of screen are 4 more examples of complex mineral samples and how nanosims can inform their chemistry. They also demonstrate how electron micrographs and nanosims data can be combined to improve sample characterization. Can I get the next slide, Ollie? So another application of nanosims related to critical minerals is analysis of phytomining, particularly those elements at low concentrations. Put simply, some plant species can accumulate massive concentrations of certain elements, including several of the critical minerals, and these plants can be harvested with the elements of interest easily extracted. One particularly well-studied example is the small yellow-flowered Elysium, which has been shown to accumulate nickel at levels approaching 3% of dry weight, which is orders of magnitude greater than what we would consider normal. In nanosims analysis by Smart & Co., nickel accumulation was shown to be prevalent in the trichomes, or the small hairs of the Elysium leaves. And these are shown in the secondary electron micrograph in the middle of the screen. In the typical nanosims data output on the right of the screen, we see collected images representing, firstly, in the top right, an SEM of the sample, which shows the structure. And this is useful to locate what structures are actually being analysed for each analysis. Next to that, to the left, is a 12 carbon 14 nitrogen map. And this is a proxy for nitrogen chemistry of the sample. And it's also useful for showing sample structure. Below that, we have data for the 2 main isotopes of nickel, both 58 and 60. And interestingly, the authors measured both the negative nickel isotope ions in the second row there from the top, and also the positive ions at the bottom there, which is another feature of the nanosims, being able to measure both positive and negatively charged ions. So all of these nanosims images are simple heat maps with red indicative of high counts and black representing absence of the ion species of interest. Again, this data highlights the potential of nanosims in these types of studies. Grab the next slide, Molly. So the final application of nanosims in the study of critical minerals relates to its use in microbiology. What I found startling when I was preparing these slides was just how little work had been done in this space. To me, it was an obviously good marriage of technique and scientific question. Furthermore, when I put the terms into Google, I was bombarded with articles discussing the potential of microbial processes in recovery of critical minerals. So obviously it is a pretty good fit, and many others have thought that as well. So this lack of nanosims application is surprising, as like I've shown, nanosims can easily separate and measure all the critical minerals, including their individual isotopes. Nanosims also works at scales that are relevant to microbiology and has been employed many times to demonstrate other chemistry at single cell and subcellular scales. It's very easy to see how any critical mineral could be observed in a single microbial cell, and I envisage generation of images similar to the image at the bottom right of screen, where we have a single microbial cell with numerous discrete features, little black dots, representing concentrations of chemistry of interest. So the take-home message here for analysis of critical minerals with something like nanosims is potential, and that's where I guess you guys come in. And I hope I've shown some simple applications of how nanosims could be used in the study of critical minerals. And I encourage you to reach out if you have any interest exploring this method any further. And I'm going to leave it there and pass it off to Danny, who's going to talk to you about some APT applications. Now, you can skip over that slide, Holly.
Hello, everybody. My name is Danny Perea, and see if I can take the screen. There we go. My name is Danny Perea. I'm a material scientist here at EMSL, and I'm going to talk along the same kind of theme of a high-resolution characterization technique that can be applied to critical minerals. And specifically, I'm going to be describing this technique called atom probe tomography, which is one of the many capabilities being described here today. So what is atom probe tomography? Well, it's a 3D imaging mass spectral technique that provides information about the 3D composition of a material from the near atomic to the nanoscale. And so at the bottom left here is an image of an atom probe, or the atom probe we have in EMSL, and it has the capability to map with sub-nanometer spatial resolution and on the order of about 10 part per million at best sensitivity, the 3D atom by atom composition of a material. And so the way it works is atoms or specimens are prepared, and I'll describe that in the next slide, into needles shape morphology, and an electric field is applied between what's called a local electrode and the specimen that creates a high electric field high enough with the help of a laser to ionize or remove atoms from the surface one by one. Now, this is all in an ultra-high vacuum chamber. Those ions travel and hit a position-sensitive detector. Now, the pulsing of the laser also starts a clock, so it's a time-of-flight technique similar to what Jeremy had just described as the TOF SIMS measurement, not necessarily the nanosims. And so from that time of flight, we get the mass. So we get a mass spectrum. So shown on the right here in the upper mass spectral image is an example of a silicon-rich material with some aluminum. And not only can we map the individual silicon aluminum, we can map out and identify with enough spatial resolution or enough sensitivity here, each isotope of every element, right? And each of these counts that make up the histogram in this mass spectra, we not only know the time of flight or the mass, but we also know the 3D position. So from that information, from the mass and the 3D position of every detected ion, allows us to reconstruct in 3 dimensions a composition point cloud map, which is shown in this movie example here, where you can see aluminum atoms. I've chosen to ignore the silicon atoms and just looking at the aluminum atoms, which are in green. And so having that 3D distribution is really powerful because it can tell you how those atoms are distributed. We can see in this case, they're distributed into these clusters, which we can quantify. We can measure composition profiles along any arbitrary direction. And so this information is really useful for understanding how different elements, and aluminum can be substituted out for any critical element there, how they're distributed within the material or a mineral, right? So that's how it works. And in this technique, the sample itself is the primary optic of this of atom probe tomography, and so we have to create these needles, and we do that in a what's called a focused ion beam capability that can be built into the scanning electron microscope that Odeta described a couple two capabilities back. So within that same instrument, what we can do is we can literally cut out a region of interest that we've identified in the SEM here. We can mill away material and lift out very small micron size, so microscopic volumes that are targeted from the material that have been identified with the SEM, right? We use the SEM to identify regions of interest, and we can pick them up, we remove them, pick them up, and we populate these micropost arrays with a portion of the sample, right? And if we turn that sideways, we can see this little piece of the material we took out that we removed, and using an angular milling technique from the top with the ions, we can sharpen this bulk sample into this needle. And ultimately, we want to create a needle morphology with the tip diameter that's on less than about 100 nanometers. And so we can be clever in that we can prepare the samples not only on these micropost arrays, which are just these silicon chips that are normally going to the atom probe, but we also have the capability or the ability to prepare them onto TEM half grid, so special substrates that are compatible not only with APT, but transmission electron microscopy, which is a extremely high resolution, atomic level resolution imaging technique with electrons that Odeta had described. So we can do one-to-one correlation with atom probe tomography. So very complementary analyses. Okay, so now I'm going to describe 3 kind of applications of atom probe tomography that can be related to critical mineral and critical element research, and they're along 3 very distinct kind of types of science. And this is trying to understand one here, which will describe soil matter interaction. So looking at how critical elements are distributed in soils and how they integrate or interact or associate, better word, associate with different types of minerals and different types of organics in the soil. I'll talk about another application of being able to use APT to look at biominerals. So the potential for both plants and organisms to sequester critical elements and incorporate them into minerals or into soil tissues or plant tissues like Jeremy had described. And then another application where we're just going to be looking at the mineral itself, just targeting critical element bearing minerals like Odeta had described, and being able to describe at the near atomic scale their distribution. So we can identify them and how are they associated within the mineral there. So in this first example here, this is here meant to show some work we did with the previous EMSL user, where we focused on using APT in addition to electron microscopy to understand how heavy metals are distributed throughout a material. And in this case, silver. So on the bottom or the top right up here, this is a scanning electron microscope image of a region of interest with EDX analysis pointing out regions rich in iron and carbon. So this is an iron-bearing mineral, which turns out to be a pyrite type of material, ferrihydrite, I'm sorry, material. And in here, with this mapping, they were able to show that there's segregation of silver. So this silver was something that was very interesting to the user. And we can see it's segregated there in green here. And so what we did was with the FibSEM, we can go in and target this region that we've identified with electron microscopy, cut out that region, make APT needles, and then study them. And so we can do that with both APT and the TEM. So here's the TEM image on the right, where we can see these small clusters of silver and how they're associated with spatially with the different types of minerals, in this case, ferrihydrite. And to get a better distribution or better higher resolution image of this kind of association, atom probe shows us that the silver, which is shown in green as these little dots, these high concentrated regions, are these individual nanoparticles. And we can see they're associated there with the iron and sulfur-rich regions, which is a pyrite type material. And in between there, we can see organic material. So this carbon in between there. So this is, and this kind of information helped our user develop a model to understand how heavy elements like silver are moving through the soil and are associating with different elements in chemistry, to understand how how silver is moving in the environment there. So it led to an important model and a predictive understanding. So down from the atomic scale, we can make inferences about how these elements are distributed in the environment. Another example here, another area of research is trying to understand how metals and critical elements are distributed in biominerals, right? Jeremy described a way of plants being able to sequester and concentrate heavy elements like nickel. Other organisms may take those heavy elements and form biominerals, right? And we want to know how that is actually occurring. How do these organisms actually do the work of biomineralization, but also characterize the biominerals themselves. So this is an example not aimed at looking at critical minerals per se, but rather it's an example of the potential for that. And so this is also another work, some work done with a past user where we were looking at these little planktonic microorganisms called foraminifera that were cultured in a laboratory setting. And we were interested in looking at metal distributions around what's called the primary organic membrane that's thought to mitigate the biomineralization, that's meant to template it. And in SEM, we can see that with this arrow that's shown here, and getting a higher resolution image in a TEM of that same sample, we can see that primary organic region there pointed in this line. And this was a very nice study where we did a nice correlation with time of flight SIMS, secondary ion mass spectrometry, a technique similar to what Jeremy had just described, where they can map out the isotopic distributions of magnesium and calcium, which make up the mineral. And so we can see these fluctuations, which were controlled in the laboratory. And what we wanted to know was what alkali elements are present there and their distribution, because they're a clue to how the protein itself is templating the growth of this biomineral. Now, the SIMS was able to tell that there's some sodium and calcium incorporation right at the magnesium is flanking on the either side of this membrane, this primary organic membrane, and sodium is right at the middle, right? But this is a very large scale on the order of microns. We suspected that was actually at a much higher resolution. So we applied APT and we were able to capture the interface using FIB-SEM to target that mineral organic interface, the primary organic interface. So we can see there the green is the carbon and blue is the calcite. We captured the interface and we can measure the composition. We sure enough corroborate the evidence from SIMS. We can see the sodium right butting up against the interface of the organic primary organic membrane, the calcite, and then flanking on side of that is the magnesium here. But the scale here is on the order of nanometers, where on the nanosims, it was because of the probe size much bigger, it's on the order of microns. So we can use this APT data to further refine the kind of information that we're getting from this much higher sensitivity but lower resolution instrument with SIMS. And again, this is helping us to understand how how organisms can sequester elements into the biominerals. So you can imagine, a study with critical minerals. And so the last kind of area of, broad area of science where APT can be applied in this critical element, rare earth element area is with looking at the minerals themselves. And so I'm drawing on some examples, not done by us, but in the literature, where people have looked at geologic materials or minerals to understand the distribution of, say, rare earth elements in them. And so This is a relatively new area, the application of APT to biominerals. And so what we want to show here is, here this happens to be a zircon, a silicon oxide material, and inside of there we can see the mapping of ytterbium and lead, and at the scale here, so we're able to detect and map that out. And in the bulk material, like an olivine, olivines are known to be a host for rare earth elements. The application of APT done here shows the detection in the mass spectrum, but not only that, we have the 3D distribution of those rare earth elements there. So this information is helping to understand, how the elements identify which elements exist, as well as their distribution, and even to quantify out their composition. So I'm going to end here with just kind of this concept of, with Odeta talking about EM and Jeremy about the nanosims, you get an idea that these different techniques have different sensitivities, so their ability to detect different levels of rare earth elements, and their spatial resolution, their ability to resolve those elements within specific features, right? And they span from the micro scale down to the nanoscale, right? And they scan, the percent ranges of concentrations down, as Jeremy mentioned, even to the part per million, part per billion region. So each of these techniques is important by itself, but when we combine them together, and I showed a few of those examples with TEM and SIM, so electron microscopy, secondary mass spectrometry, and APT, you get a much better understanding than if you were to use any of those techniques by themselves. So with that, I'm going to end here and pass the presentation on to Arunima, who will be talking about TerraForms.
Thanks, Danny. Hi, everybody. So I'll be talking about TerraForms, and maybe briefly I will give a little bit of description about what are actually TerraForms. So TerraForms are a suite of different capabilities which help understand different soil biogeochemical processes. And we can do a lot of spatial analysis on these platforms, including the analysis that you just heard about. And in soil, biogeochemical processes are very spatial, and and it's very hard to characterize these spatially because of most of the characterization techniques use homogenization of the soil. And so why is it important to visualize these in a spatial way? Is because the spatial organization in soil can be a controlling factor in many important biogeochemical processes. And biogeochemistry occurs spatially inside of these soil pores. And so heterogeneity in soil is what makes soil so diverse. And we love to study this diversity. And today I'm going to talk about how we can look at microbe-mineral interactions in the context of biomining of critical minerals. And so if we are looking at soil, we want to maintain this heterogeneity. We want to increase the measurement resolution while decreasing the complexity of soil, but at the same time maintain heterogeneity. And that's where TerraForms come in. So with TerraForms, we are able to isolate and control key processes at increased resolution and map biogeochemistry, whether looking at micromineral interactions inside of these platforms. So we can, these are microfluidic platforms and also can be printed using 3D printing. So we can mimic soil aggregate morphology. As you can see here, we can mimic soil porosities and different porosities and soil pore connectivity. And then we can also mimic the mineralogy. So the mineralogy we can mimic using certain ores that you can purchase, like for example, lithium ores, or it could be synthesized minerals in lab. And these synthesized minerals or ores, as long as these are in form of powder, we can incorporate them inside of these platforms. We can grow plant roots and microbes in there, and we can also observe them using different characterization techniques. So as I mentioned, these can be microfluidic based or 3D printed. So for microfluidic based platforms, we start with a glass cover slip, and then we put an Indian tin oxide coating on it. This makes the entire platform conductive and amenable for most of our mass spec techniques, as well as could be for SCM as well that you just heard. And then we build our microfluidic habitat, the soil aggregate structure, where we also amend the mineralogy, which is depending on what you want to study. It could be different types of minerals. And then we cover it with a membrane, and that becomes your habitat where you can perform your experiments. And so in this way, once you have performed your experiments, we can perform several biochemical assays. We can track growth of the microbes or plants, and we can collect the biomass at the end for different types of omics. It could be proteomics, metabolomics, or you could also do genomics with it. And then finally, we can image these using different types of chemical imaging. I will show 3 different types of chemical imaging that we have used on these platforms today, which will give you an idea of what we can do when we are looking at micromineral interactions specifically. And so here are some of the TerraForms designs that EMSL offers. The first one is pore-to-chip, and I'm going to be talking a little bit more about pore-to-chip. This is our most recent capability with the TerraForms. Then we also have the standard micromodels, where we have done extensive research on fungal mineral interactions. That's what I'm going to show you today. And then finally, the 3D printed habitats are the subtep and microbial platforms. I'm not going to be describing these today, but these are the platforms you can use for doing extensive plant research. You could amend these with minerals as well. And these offer you the possibility of time-resolved metabolomics. These are also very high throughput, so the analysis time is pretty short. So using these, you could look at micromineral interactions, look at biomining, biogeochemistry, screen for critical elements hyperaccumulating microbial or plant species. So these are just some example research that could be done on these platforms, some of which we are already doing, and something to think about. Okay, so I'm going to describe pore-to-chip, which is a standard microfluidic platform and could be used for mostly microbial research. So we start with a soil core, and then we scan these soil cores using X-ray computed tomography, which gives us the soil pore network connectivity and pore sizes, pore size distributions, or pore throat sizes. So with all this information, we use this information and we construct this pore network connectivity in 3D. Pore-to-chip is actually a Python code that translates all of this into a 2D design. And this 2D design then can be printed into a TerraForm. So this gives a very heterogeneous pore network, which is very with the soil pore connectivity, which is very representative of any soil core that you want to study. And these are created in using epoxy, and you could amend different types of minerals in these. like I said, these minerals, if they're in powder form, it could be added to these platforms. And we also have, I must mention that we also have connection with BER structural biology beamline at APS, which can provide us micro-CT data with which we can really focus on really small pore sizes, which also could be generated and incorporated into the design of the platform. So these are all customizable, as I mentioned, and are compatible, and the polymers are compatible with range of measurements. But to measure any kind of biogeochemistry, we must have a workflow, and we have developed that workflow for sample prep. So as I mentioned before, we design the platform, we perform our incubations, be it microbial or plant incubations. And then at the end of the incubation, and throughout the incubation, we are able to non-destructively track growth using optical microscopy. But at that point, when your incubations are done, we freeze, lyophilize these samples for our different in situ measurements. At which point we can either collect all the microbial or plant root biomass for omics, or we can remove the top membrane and then pass on the sample for in situ chemical imaging. And so we have collaborations with several different synchrotron facilities, one of which APS is part of our ficus, and you can request both EMSL and APS beamline resources as part of these the proposal. And so we do the incubations. We could do the incubations here, or you could do it in your lab, and send us the samples. We prep the samples. The first sample analysis could be our mass spec analysis, and then the sample gets separated for biomass and minerals, and where we can do different types of elemental mapping, as you can see here at beamlines. So here is an example of tracking metal transport in fungal biomass. In this case, the fungi was grown inside of these micromodels, and we looked at different metals. I'm showing an example of potassium transport inside of the fungus, where in presence of the minerals, the fungus was extracting a lot of potassium. And so usually fungi does this using exuding a lot of organic acids. The next analysis was to look at if there was organic acids present inside of on the micromodel surface. And so we mapped different organic acids on the micromodel surface produced by the fungi. And so these organic acids are used to chelate and uptake different elements inside of the microbes, and that's how they biomine and accumulate these metals. So that we could track using X-ray fluorescence and XANES at SSRL, which is a beamline facility. You could do this at APS as well. As I mentioned, APS is our ficus partner, so this analysis is also available to you when you collaborate with them through a ficus proposal. And so in this case, we mapped the potassium citrate inside of the fungal hyphae, which shows that the fungus was uptaking the potassium using citric acid. So it could be applicable to any other metals. As an example, I'm showing here what could be done. And these are all done on the same sample. So the same sample first gets analyzed using mass spec here, and then it will be transferred to our ficus partners. And so in this way, you could develop an entire workflow. You start with the TerraForms pore-to-chip design. You make a chip from a soil core. Then you perform your incubations. You could do secondary ion mass spectrometry, MALDI, and then transfer for looking at speciation or the chemistry of how these biomining processes are occurring. And then ultimately, you could also look at changes occurring on the metal, so on the surfaces of the micromodels themselves. And so finally, I will leave with one last slide where these port-to-chip micromodels are now being generated in a really high throughput manner. We are conducting field studies in our local field site with different mineral amendments, and so that's also being made available to EMSL users. So that's also a possibility. And so this is my acknowledgement with different collaborators. I would love to make you part of this slide as well. So please contact me for TerraForms related work. Thank you.
Thank you, Arunima. I'm going to invite everybody to come back on screen so that we can answer a few questions. We have everybody. All right. And just for everyone in attendance, we do have a bunch of questions. We're probably going to just take 3, and then I'll have our speakers follow up with you via email so we can get you out of here at 1. So first question, I'm going to actually direct to Odeta. Is the exploratory call specific to looking at critical minerals, or could we propose something focused on biominerals as presented by Danny Perea?
For that, I will say that you should look at the proposal details. I know that we would like and encourage people to submit proposals with respect to critical mineral research, but there are other directions that are in the exploratory proposal, and you are absolutely welcome to submit proposals in those directions.
Great, thank you. And we did include the link in the chat if you're interested in looking at the exploratory call. Letters of intent are due July 2nd. Question for Arunima. Arunima, can TerraForms 3D print manganese oxides into different structures? How thin is the TerraFilm and how large is the platform?
Yes, you could do that, absolutely. The channel depth, we can control it, but it's usually in the micrometer range. I would say like 30 micrometer and then going up to like maybe 300, 350 micrometer. I would not recommend going beyond that size range for any kind of microbial or plant incubations.
Okay, thank you. And we'd encourage you to follow up with Arunima if you have any specific questions related to TerraForms. Okay, let's see, Jeremy. Jeremy, have you tried nanosims to detect other elements except for nickel and other hyperaccumulators? Because other hyperaccumulators can also accumulate other metals.
I personally haven't tried it, but I have no qualms that it wouldn't work. It would certainly work. Whether it's nickel, magnesium, arsenic, it doesn't really matter to me what it is. As long as I know what I'm looking for, I will be able to find it if it's there in any sort of abundance.
Great, thank you. Danny, here's a question for you. Would EMSL expect an exploratory proposal to incorporate several correlative analyses for a single question, opposed to just using SEM or APT?
It certainly helps. One can absolutely submit a exploratory. Those are meant to be kind of smaller projects. So if it's an area of science, you know, if the scope is such that only one instrument is necessary, then yeah, that can be done. But internally, what we'll do is we'll look at that proposal anyways between us, and if we see opportunity to expand that with the other techniques, we tend to make those recommendations. But I would encourage you, the user, to go ahead and submit, and maybe at least contact us directly to help them develop that proposal idea.
Great. One other question for Odeta. Odeta, what is the difference between EMSL SEM and a regular SEM that we would have in other national labs or universities?
So there will not be any difference if you have all the detectors that we have here, if you have the same high resolution that we have here. the SEMs as instrumentation have developed tremendously in the last 10 years. And so if your instrument has all those capabilities that you need for your research, they're not going to be any different. But the only difference that we have here is opportunity to include their different techniques, like Danny and Jeremy talked, and that is something, that's the power actually of EMSL capabilities. The fact that we have all these different techniques in one place, and you have the ability to use them all in one proposal.
Great, thank you. All right, with that, I'm going to close today's webinar, and any remaining questions, we will have our speakers today reach out with answers to you directly, or we encourage you to email each of them with any additional questions. So thanks for joining us. We hope to see you back on June 25th for our next webinar. Have a great day.